advance post-plant resistance to chestnut for the chestnut blight. Another example is for plum pods for protecting it against Charcot disease. There's a product now that is starting to be reviewed by FDA that looks at trying to make it so that if you take an apple and you cut it, it starts to brown, starts getting dark. You can keep it looking fresh by cutting out phenol oxidase activity, and they do that by focusing on the gene and the gene patterns, the gene pathways. They take the genes out and basically kind of turn them around and put them back in, and it stops that pathway. Is that, I don't know if that's going to be opened, if that's going to be picked up by the market or not. So, you know, all I've tried to do in this first half of the talk, and I'm going to have to really move quick, is to make you realize that insect-resistant genes, they're out there in a large fraction of a small component of our agriculture by diversity of species, but a large fraction of our acreage. There's plenty more, and I just wanted to open that question of who chooses and how. Maybe we can return to that. What I'd like to do in the next 20 minutes is point out how these things have affected our population dynamics of insects and population genetics. Realize that I'm just talking about the insect-resistant transgenes. There's the herbicide-tolerant things. There's a lot of other components that we could talk about when we talk about GMOs in agriculture, but I'll just focus in on this part. So this is a part that Tim got involved with. So with European corn borer, this is an insect that overwinters as a late instar larvae and quickly turns into a pupae in the spring. Here you see it as a pupae in the spring. Emerges as an adult with males and females. Lays its eggs, they hatch, there's 5 larval instars, and the cycle repeats usually one time and then it goes through the winter. There's your life cycle of the European corn borer. Here we are in the United States of America, humongous grower of corn, and along comes, where do you think the European corn borer came from? Help me out. Europe, yes. It's an invasive species. European corn borer showed up around 1920, showed up again around 1950, came from Europe. And so with an invasive species that's dramatically influencing our agriculture, people start trying to manage it and monitor it. And here's monitoring data that was coming from the upper Midwest. Here's the number of corn borer larvae per 100 stalks late in the winter, sort of getting an idea of its population density. And you see these cyclic dynamics, up and down in these cycles. BT corn was introduced on the downslope of the cycle. The population is going way down. You might think they'd climb back up again, but they didn't really get very far, and then they kind of went, not going very far. Tim actually got involved from some independent work he was doing, not with me. He was doing some work out in Wageningen in the Netherlands, and he modeled this out and figured out how to estimate a time series, showed that these really follow a time series, a certain frequency. There are 7-year cycles, 6 or 7-year cycles. What that led to understanding and looking at this in multiple states. There's Minnesota, Illinois, Wisconsin. You see some cyclic dynamics. And you can see the dynamics of the insect. The shaded area is the percent of acreage in BT corn rising, starting from 1996, 97 on. So do we have, is one influencing the other? Here's the moving average using Tim's estimate of what the frequency is, so you can kind of smooth it out. Get an idea of the moving average plotted as a function of the percent of the acreage that's in BT corn. You can see dramatic effects. If you represent it as a cumulative acreage over the last several years, you can see a very dramatic effect happening in a sort of a threshold sort of response. And I think that's what's really happening here. Is the insects are being exposed to this BT. It's knocking their population down, and it's happening generation after generation. So we get some nice classic, think about this, you've got cyclic dynamics. Every time the population rises, gets high enough, it's going to start crashing again. When it's low, it starts rising again. So if remember that, you know, the numbers that you have at time t plus 1, in other words, next year, compared to the numbers you have at time t this year, if the numbers, you know, if you get more next year than you have this year, you have an increase in the population. If you have less next year compared to that, you're going to go, you know, it's going to be a decrease in the population. So, nice classic scenario where the numbers of time t, or the numbers of t plus 1 over the numbers of time t, that's what we call the intrinsic rate of increase. And if we plot this intrinsic rate of increase by the growth rate, when you have cyclic dynamics, you expect something like this. When density is low, growth rate is high. Populations are low, nothing's stopping it from growing, so their growth rate is high. When the density gets high, something's stopping it from growing. Maybe it's a pathogen. That's what I think it is. There was a multitude of possibilities. One simple possibility is you have a pathogen that's being transmitted among the insects. Population is high, epidemics occur, growth drops. Populations start scattering about because populations are low, pathogen has a hard time, growth builds up back up again. So you expect this kind of pattern, inverse density dependence, when you have these cyclic dynamics. And that's basically what we saw. Here's your growth rate in Minnesota. The open circles are the growth rates that were occurring over the last 30, 40 years, before BT. After BT, these different color circles, you can see that the growth rate is still following this pattern, but we've shifted the whole process down. So we're at a large scale, statewide scale, we're dropping the populations down by adoption rates of this transgenic technology. It's really just pretty simple. In the absence of BT corn, European corn borer flies around, lays its eggs everywhere, and populations grow or fall. In the presence of BT corn over here, whenever the population, whenever a female lays her eggs on the BT plant, nothing emerges. It's a sink. You get these source sink dynamics. And people called it this halo effect. And we were studying this because as entomologists, we knew that this is nice to see the populations drop of a pest species, but it also creates a tremendous selective pressure. It's going to influence the genetics of the population. It's going to make it so that we're worried about resistance cropping up. And what I'll emphasize a bit more later is there are management plans put in place to try and minimize or slow the rate of resistance building up to this process. So we have refuges. We have areas that are planted to non-BT corn, and they're mandated so that growers have to put a certain amount of acreage in non-BT crop so that we can preserve some susceptible phenotypes in the population. We get this moving back and forth, but keep the susceptible phenotypes there. And we wanted to encourage growers to do that. So we studied this and tried to show that there's an economic advantage to keeping that non-BT acreage in the environment. This shows data from Minnesota, Wisconsin, Illinois, and over here, Iowa, Nebraska. Up at the top, you'll see the annual economic benefit in millions of dollars. Up top, both of these columns, this row up here, you see over time in different states what was the millions of dollars of benefit or 1 year loss to that hectare, to the acreage that was in the BT crop. But if you look at the non-BT hectares, they were gaining quite a bit more. The scale over here goes from 0 to 175. The scale here goes from 0 to 250. So we're actually gaining more value in the non-BT hectares planted nearby because they didn't have to pay the seed cost. And so we're trying to convince them, you know, hang on to these refuges, and that's why we did that. That's why we did that study. Here's the cumulative benefit to the BT hectares. We'll just look at these states here. To the BT hectares, it's in red. To the non-BT hectares, you're seeing it a lot more. And then the blue is the total. I don't know what these yellow things are here. On my slides. So through these source sink dynamics, we're dropping the population quite a bit, and it was surprising to us to actually be able to tease this out, because European corn borer has about 200 host plants. For us to actually see this on a large regional scale in the presence of an insect that's choosing among BT and non-BT corn plants, but also scores of other plants in the environment, was pretty exciting for us. We'll go back in time a little bit and consider some of the other pests. In corn today, maize today, this is the whole range of species that are being targeted with various transgenes and vectors and stacks. The other big crop that we're dealing with that's influenced by BT technology is cotton. And we've got a bunch of different lepidopterans, caterpillar species. Let's take a look at this one, pink bollworm, because it's a specialist. Has to feed on cotton. And we're going to go to the desert southwest, where we're in like Arizona, center pivot irrigation. You might have 200, 500 acres under center pivot irrigation, and outside of that, it's desert. So this species is feeding right in that irrigated area. And you can just ask the question, what happens to the density as pink bollworm moves among these fields? So the same equation that you looked at before. And here, these Carriaridol in 2003, predating what I just showed you by almost by 8 or 9 years. Just to ask, what would happen in a single field as populations would grow or fall, and then move among BT and non-BT fields. And it's really a simple development off this equation. We're going to take populations next year as a function of the growth rate, and the population this year, minus the emigrants, the ones that flew out of that field, plus the immigrants, the ones that flew into that field, but only if they flew in from a non-BT field. If there was a BT field nearby, nothing comes out of that. So we're going to just add immigrants from the non-BT fields that are immigrating into that particular field. Imagine 100 fields. Within the one that you're looking at, you just have growth rates dependent on intrinsic rate of increase, and then you have some that emigrate out, some that immigrate in. There's your math. That's all there is to it. Nice simple equation. Here's your numbers. Next year, depending on the numbers this year, and the growth rate, minus the ones that emigrate. So we just take the growth rate in that field, and we add in a term, E, emigration rate. Some fraction is going to fly out. There's your immigrants. They go away. All of this is your immigrants. Just one term, and you just have how many other fields you're looking at, the number of fields minus the 100 fields. So you take 100 minus the one that you're in, so you've got 99 fields. The proportion of them that are not BT, the growth rate inside of those non-BT fields, and get rid of the ones that emigrate out, and that the fraction that emigrate out are the ones that can immigrate in. This whole thing divided by the total number of fields, get an average contribution per field into your field that you're looking at, and you can see that depending on your net reproductive rate, here's 4 of them drawn out here, and here, and depending on your emigration rate, the rate at which they fly around, you'll end up with some kind of curve, and if you're below this curve in terms of immigration rate or proportion BT cotton, your density will increase. But if you're above this curve, density will decrease. You can almost see an asymptotic threshold sort of response. For example, if you're gaining 11% per year in your growth in your population, as long as you were, say, an insect that had 40% of it, that 30 or 40% that flies out, as long as you get to a point where you're up around 30% BT cotton, you're going to start driving populations down. So they're doing it. It is happening there as well. I always like to throw a little population dynamics in there. This is the sort of the extension part of it. What's happening with pink bollworm in the U.S. is this technology is so successful, it's creating an eradication program. They're basically pushing pink bollworm out of the U.S. and out of parts of Mexico. And they use the BT cotton allowing 100% use, which means they relaxed the resistance management plans. They had a pheromone-based mating disruption thing, a sterile insect release, which acts to bring their susceptibles back into the population, and insecticides, and we're down to very few pink bollworms in that system. Number of insecticide applications in Arizona and what they were targeted against. This was the year '96 when BT cotton came into play, and overall you can see a dramatic increase in insecticide use. There's been a switch. There's been more for some species, but overall less. Biological control has increased a lot because of that. It's not just happening in the United States. Here's examples from China, looking at the population dynamics of a different species, as cotton bollworm. And here's the density in cotton. You can see a decrease over time. And here's the density in corn, peanuts, soybeans, and other vegetables that are grown in this highly diversified, small hectare kind of situation in China. Because it was only BT cotton, it was only in cotton that you had the transgene, but it was influencing populations in all the crops nearby. So what I've tried to do so far, in a real quick nutshell, is to show that there are insect-resistant transgenes. There's a lot, but there's just a few that we're using now, but you gain a sense that we could use a lot more. And I just showed that they can have regional impacts on population dynamics. I'll cover a little bit on what they might do with population genetics, and then just bring up some issues and go for some time for conversation. So, management of BT crops. As an entomologist, I'm actually kind of proud to be able to state this. It's the first and only proactive, legally mandated, required management plan for resistance in all of agriculture. And if I'm not mistaken, all of medicine as well. If you have a new antibiotic and it gets out on the market, concerns about resistance are not going to be surprising to you, right? I mean, you've got resistant bacteria all the time. Who manages that? You know, if people talk about what we should or shouldn't do, is there any legally mandated thing? I mean, in agriculture, if you're using a transgenic insect-resistant crop, not a weed-resistant crop, and there's discussions about why that might happen, and we've got a lot of resistant weed problems now. But it's actually required, and it's required by EPA prior to the deployment of the transgenic crop in the first place. So we have it out there. It's complicated, and it's not always working, but the concept behind it is pretty simple, and probably too simple for making it work really well. Remember, gene flow can move through selection. So here we have, you know, a population of little cute bugs, another population with a resistant individual there. You spray or put any kind of selective pressure that causes mortality. You're going to select for the resistant type if it can survive. But you also, once you get down here and get selection, you can also get movement among fields, so you get dispersal. So we got gene flow running through selection and dispersal. And what we have to do when we deal with agriculture today, and you know, farmers, I sit there and talk to them about this stuff, we have to cover resistance management. Before transgenic crops, I was covering all these things, you know. You have to get comfortable with homozygotes, heterozygotes, dominant recessive alleles, gene flow, so forth. And actually today, if you're working with insecticides, it boils down to the IRAC codes. Insect Resistance Action Committee helps define what these codes are and helps define how you manage various insecticides. There's an IRAC, there's an FRAC for fungicides, there's an HRAC for herbicides. But on top of all this now, we have to deal with all of these terms as well, and obviously I'm not going to have time to cover them all. But there's a couple that are pretty interesting from the point of view of population genetics, ones that we have that you may not think about in other parts of biology. I'll talk about a little about structured, non-structured refuge, functional dominance. I think that's about as far as I can deal with today. But functional dominance, you know, you think of an allele and whether it's dominant or whether it's expressed in the heterozygote state, right? In the case of Bt crops, the concept of whether that R allele, that resistant allele, compared to the S susceptible allele, depends on the dose. So whether the gene is dominant or recessive, whether it's expressed in the presence of the crop in the heterozygote state, relates to the dose of the insecticide itself. So if you have a Bt crop, how much protein is it expressing? And that changes over time. Anytime you grow a plant, and you've got ones that taste real good because they're. But if you have a plant, you can tell whether plants are looking really good and look real nutritious. They're expressing a lot of certain proteins, or ones that as they senesce or get old, or they change, they just senescence through their growth stage. That expression changes over time. This is what it kind of looks like if we have a susceptible percent killed. Susceptible homozygote, resistant homozygote, heterozygote here. Put the insecticide dose here. Obviously, the resistant heterozygote is surviving. So the R allele now is dominant because it's being expressed. All we have to do is change the dose up to here, and now the R allele is recessive because it's not being expressed. And what we basically try and do with the simplest case of, and the first set of cases of a priori deployment of resistance management crops, is make sure that we have a dose that we call a high dose, and by that we're defining, PA typically defines as a dose way up here. It's going to kill, it might kill the homozygote recessive lepidopterans, but even as the plant senesces, it'll kill off almost all the heterozygotes here, so it stays as a high dose. It's mandated to be deployed prior to, at the time of commercialization. So if the product is effective, then most of the population is carrying susceptible genes, right? So it's being deployed when the resistant gene is extremely rare. And we actually get to start measuring it. It's like 1 in a million. You get a resistant allele out there. So you deploy it when there's 1 in a million insects that are resistant. That 1 in a million might survive in the crop. But in the meantime, you've got this little refuge patch out there, 10% of the acreage, say, and that crop is sitting there just boiling with susceptible phenotypes. So you got 1 in a million that survive, and a whole bunch of susceptible phenotypes. They mate, and all the matings from them will be heterozygotes. So now those heterozygotes get killed because the dose of the plant is being kept up at a high dose. That's what we call a high dose refuge strategy. It's not perfect. It assumes a lot. It assumes, single alleles carrying this resistant trait, not a quantitative kind of polygenic process. It assumes that the ecology of the insect is going to operate so that you get random mating from the susceptible plot, from the refuge plot. So there's a lot of assumptions, and you try and make these assumptions work for multiple species. It gets complicated, but here's kind of what we have to do. And plant expresses a high dose, refuge provides susceptibles. We get immigration of susceptibles into the area where the rare resistant type survives. Random mating, that'd be interesting. That the heterozygotes are killed, excuse the typo. And it started when the R allele frequency is very low. It's mandated at the time of first introduction, and that's one of the things EPA does. So, how's it working? Here in 2013, Tabashnik et al. Put this out where we now have lessons from the first 1 billion acres of growing these crops. In 2005, there was 9 species who was able to look at gene frequencies for the R allele, and all but one of them had stayed in a susceptible phenotype, and one of them out of 9 had reduced efficacy of the Bt crop, so we're getting some resistance. By 2010, we had 13 species looked at in 24 different populations, and you can see we're getting some resistant types. We're getting that fraction out of 24 populations that are getting reduced efficacy. 1 to 6% was truly resistant, didn't have anything between the two. And then these are, the gene frequency had changed, but we couldn't see any change in the efficacy in the field, and the rest was susceptible. So it's not working perfectly. We're closing in on 2 decades, and we're continuing to have to deal with this problem. So we're having, I have to skip this slide. So we are having impacts on the population genetics of the plant, and we shouldn't be surprised. Here's one example for what's called the Hessian fly. This little insect here was an invasive species when Hessian soldiers came over in the Revolutionary War, and they infest wheat. And the deployment of insect-resistant germplasm to manage Hessian fly over the last, I think, about 50 years, over 6 wheat cultivars have been released. They're constantly breeding for resistant phenotypes. They have to keep releasing new ones because the insect keeps developing resistance. And the pest has evolved over 16 biotypes. And we have management programs that involves variable spatial deployment of resistant germplasm. So entomologists are pretty well trained in the idea that we have to deal with resistance management, and we kind of got that as part of the way the U.S. and most of the developed world deals with deployment of these crops. So I'll try and summarize by saying that there's I was talking about the weed crops. I'd be dealing with something else, but for the insect-resistant crops, insect-resistant transgenes, it can improve farm worker safety. Most of the debates I see that are blasted all over the news, and New York Times, etc., you almost never see even a phrase about the farm workers in the whole scheme of things, but it's farm workers that I interact with when I'm dealing. Most of the settings I deal with are small farms, small family farms, and the family are workers. It's scale neutral. It influences the benefit to the big farms as compared to the small farms about equally. I mean, it's not benefiting one scale versus another. That's not the same for the herbicide-tolerant crops, where it's more tied to labor issues. You do achieve management of the targeted pests. You can manage insect vector viruses, which is pretty cool for entomologists. We get regional suppression, regional benefits, area-wide management, local eradication, increased biocontrol, reduced insecticide load. This is just a classic IPM, integrated pest management triangle. These techniques that we've developed here are all trying to avoid the pest problem, and at the same time we develop sampling and monitoring programs, and when populations exceed thresholds, then we get involved with using chemicals and using them carefully. So are the Bt and viral-resistant GMOs pesticides, like up here? That's how EPA regulates them, as pest-incorporated pesticides, HIPs, or are they host plant-resistant genes? Both. We really don't have a regulatory framework to deal with that. We're going to have 9 billion people here in the next 50 years or so. How are we going to keep the increased population well fed? That becomes an issue, and it interacts with this GMO thing a lot. This is way back in 2013 or 2003, where this man here broke into a place where they were holding food aid. It was a large quantity.